Assalamu alaikum dear students, welcome to your channel The Anatomy Canvas. In today's class of Histology Foundation Module Lecture 3, we are going to discuss the cell histology. So the learning objective for the today's class are to describe the structure of the nucleus, to describe the nuclear membrane, the nuclear pore complex and nucleoplasm, and to discuss about different types of the chromatin. So first, the cell nucleus. The nucleus is a membrane limited compartment that contains genome, the genetic information in the eukaryotic cells. The nucleus is the headquarter of the cell. It is most obvious organelle in any eukaryotic cell and appear as a large dark spot in eukaryotic cell. It controls all the cell activities. So in this diagram, in this slide, you can see this is the structure of the cell, in which you can see the central dark staining area, which is the nucleus. This is the membrane limited compartment. It controls all the activities of the cell. It is a membrane bounded organelle which houses most of the genetic information and the regulatory machinery that is responsible for providing the cell with its unique characteristics. Although most cells have the single nucleus, some eukaryotic cell types have no nucleus and other have many. So anucleated cell, the example is the erythrocyte or the red blood cells, and polynucleated or multinucleated cells like many protozoans. So in human, the myocytes become polynucleated during development. So these are the different examples of the cells having either single nucleus, or they are anucleated or the polynucleated. So what about morphology of the nucleus? They usually spherical or oval shape. In these slides, in this diagram, you can see the different shapes of the nucleus, may be spherical in shape, may be ovoid in shape. Our shape may vary in different type of the cells like eosinophils, basophils, etc. The shape may vary according to the shape of the cell. So first, a little bit about the functions that the nucleus, it shows compartmentalization because of the nuclear envelope which is present, and it separates the compartments of the nucleus from the cytoplasm. It stores a cell's hereditary material, the DNA. It is the site of the DNA replication, site of DNA transcription to messenger RNA. It is the site of the ribosomal formation, and the nucleolus, the RNA and the protein required for ribosomal synthesis. They are present in nucleolus. It coordinates the cell's activity, which include the growth, the intermediary metabolism, protein synthesis and reproduction, that is cell division by regulating the gene expression. So these are generally the functions of the nucleus. So now we move on to the structure of the nucleus. Nucleus is enclosed by the nuclear envelope. Here you can see this is a nuclear envelope. It encloses the nucleus, and this nucleus contains different things like the chromatin which is present in the nucleus, the nucleolus which is present, the nuclear envelope, and the nucleoplasm which is present inside the nucleus. So these are the different components of the nucleus. We discuss all of these one by one. Here you can see a section of the nucleus in which you can see the nuclear envelope, and it consists of the two layers. The outer is continuous with that of the rough endoplasmic reticulum. In the center you can see the nucleolus is present. Also you can see the chromatin which is present. Over here you can see different types of the different nuclear pores are present in the nuclear membrane. Another slide to show you the cell. This is the structure of the cell having different vesicles and subcellular organelles, and in the center you can see the nucleus is present having many nuclear pores in it. So what is the nuclear envelope? The nuclear envelope completely encloses the nucleus and separates the cell's genetic material from surrounding cytoplasm, serving as a barrier to prevent macromolecules from diffusing freely between nucleoplasm and cytoplasm. So very important, nuclear envelope, it is responsible for compartmentalization. It separates the genetic material which is present inside the nucleus from the cytoplasm, and it also allows the movement of the certain molecules and restrict other molecules. So the nuclear envelope acting as the selective, having the selective permeability for different macromolecules. Nuclear envelope is formed by the two membranes with the perinuclear cistern space between them separates to the these membranes separates the nucleoplasm from the cytoplasm, and having the 10 to 50 nanometer the cisternal space in between these two layers. The outer nuclear membrane is continuous with the membrane of rough endoplasmic and is similarly studded with the ribosomes. So this is the diagram. Here you can see these are the two nuclear membranes, the outer nuclear membrane and the inner nuclear membrane. The outer is continuous with that of the rough endoplasmic reticulum, and like RER, this outer nuclear membrane is also studded with the ribosomes as well. The inner nuclear membrane is attached to the inner lamina, which is formed by the intermediate type of the filaments forming the lamine, forming this intermediate lamina, which is acting as a scaffold and which helps the inner nuclear membrane and gives it supports. Where these two membranes fuses with each other, there is a pore called the nuclear pore, and in the nuclear pore you can see the nuclear pore complex is present. In the nucleoplasm you can see the heterochromatin is present. This is again electron microscopic picture of the nuclear envelope. In this you can see two layers of the nuclear membrane which are present, and here you can see the nuclear pore complex is present, and in these areas you can see the heterochromatin which is present. So the space between the membrane is called the perinuclear space, and it continuous with the rough endoplasmic reticulum. So this is the space. This one is the outer nuclear membrane, this one is inner nuclear membrane, and this is the space called intermembranous space or cisternal perinuclear cisternal space, and this space is continuous with that of the space of the cisternae of the rough endoplasmic reticulum. Next is the inner surface of the nuclear envelope is bound to thin filamentous network, the lamines polypeptide. Here you can see this is the lamina which is formed by the lamine polypeptide. These are intermediate type of the filament. These are giving the mechanical support to the inner lamina of the nuclear membrane, and it also serves as a site of attachment of the chromatin fibers. Here you can also see the chromatin fibers are attached to the nuclear inner lamina, and here also you can appreciate the attachment of the chromatin fibers to the lamina. The mutation in laminin genes are responsible for several distinct human diseases, like a rare form of the muscular dystrophy, in which there is a mutation in the gene forming the lamin, and in such cases the inner lamina is affected. So in this diagram, also a beautiful diagram in which you can see the inner nuclear membrane and outer nuclear membrane. Where the two membrane fuses with each other, you can see nuclear pore is present over here. The outer membrane is continuous with that of the membrane of rough endoplasmic reticulum, and similarly it is studded with the ribosome. Between these two membranes you can see the space which is the intermembranous space, a cisternal space, and this space is continuous with that of the space of the rough endoplasmic reticulum. On the inner aspect you can see inner membrane is supported by the nuclear lamina. This is the inner lamina which is present, giving a scaffold to the inner layer of the nuclear membrane formed by the protein called the lamine protein, and the center you can see the nucleolus is also present, and here you can also appreciate the chromatin is present. In this diagram, this is the electron micrographic picture prepared by the quick freeze deep etch technique showing the nucleus, and in this also you can appreciate the two layers of the nuclear envelope, and these are then continuous with that of the rough endoplasmic reticulum, and here also you can see the nuclear pore is present. So nuclear lamina. The nuclear lamina is formed by the intermediate fibers. Here you can see this is the nuclear lamina or inner lamina. It is formed by intermediate type of the fibers, and it lies adjacent to the inner lamina of the nuclear envelope. So this nuclear lamina is the thin electron dense intermediate filament. Filamentous network like layer resides underneath the nuclear membrane. So here you can see, you can appreciate this lies underneath the nuclear membrane. In this area you can see the nuclear pore complex, and here you can see DNA forming the chromatin. This is the part of the nucleolus. In this diagram, this is the electron micrographic picture again of the portion of the nuclear and lamina, and this you can appreciate the inner lamina which is present over here. So, nuclear lamina, in addition to supporting, are the nucleoskeletal function. It is essential in many nuclear activities such as DNA replication, transcription, and gene regulation. So, different, as we have discussed, that the different heterochromatin are also attached to this lamina. So, this lamina not only acting as a scaffold for the inner lamina of the nuclear envelope, but it also help in many essential activities of the nucleus like DNA replication, transcription, and gene regulation. Another important point is that in cases when the nuclear envelope is disrupted because of the exposure to detergent or any other thing, then even then the nuclear lamina remains itself, and it maintains the shape of the nucleus. So nuclear envelope has an array of the opening called the nuclear pores. Here you can see these are the different nuclear pores which are present. At numerous sites, paired membranes of nuclear envelope are punctuated by 70 to 80 nanometer opening through the envelope. So this is the size of the opening. It is 70 to 80 nanometer opening. This is again your MCQ as well. These nuclear pores are formed by merging of inner and outer membranes of nuclear envelope. So here you can see the inner nuclear envelope and outer nuclear envelope. They merge with each other, they fuse with each other, and at this point the nuclear pore is present, which is formed by the nuclear pore complex. So what is the nuclear pore? The nuclear pores are the gateways across which movement of RNAs, the proteins, takes place between the nucleus and the cytoplasm in both the direction. So protein synthesis in cytoplasm crosses the nuclear envelope to initiate the replication and transcription of the genetic material. Similarly, messenger RNA, tRNA, and ribosomal subunits built in nucleus cross through the nuclear pore to the cytoplasm. So this diagram shows you the structure of the nuclear pore, and this nuclear pore structure is formed by the donut shape 8-fold symmetric ring shaped structures which are present at the junction where the outer and the inner leaflets meet with each other. So this is the part of the structure formed by the nucleoplasmic ring subunits, and these are the cytoplasmic ring subunits. In the center you can see this is the central framework, and this is the luminal unit which is present in between the lumen of the nuclear envelope. On the cytoplasmic site you can see the protein fibrils are present, and on the site of the nucleoplasm you can see the nuclear basket fibers are present, and these nuclear basket fibers are then given support by the terminal ring. So this is the basically the structure of the nuclear pore fibers, which are formed by the donut shape 8-fold symmetric ring shaped structures on the cytoplasmic sites. These are given the name of the cytoplasmic ring subunit, and on the side of the nucleoplasm these are given the name of nuclear ring subunits, and on the in between these two there is the central framework with the luminal subunit, and in the center you can see this is the central pore which is present. So these structures, the protein fibrils and the nuclear basket fibers, they serve to mediate the binding to the nuclear transport proteins. So again another diagram in comparison to this one, in which you can see this is the central framework. Here you can see central framework present over here, and here you can see central framework present over here. This part is the cytoplasmic ring subunits, which are donut shaped structure. You can see in diagrammatic presentation over here, and in this diagram you can see these are the cytoplasmic ring subunits, and here you can see the protein fibrils are present. In this area you can see the nucleoplasmic ring subunits with the nuclear basket fibers, and then the terminal ring. So this is basically the structure of the nuclear pore complex, which are formed by the different subunits. So nuclear pore is about 70 to 80 nanometer in total diameter and consists of around 100 proteins, which allows the free passage of the small water-soluble molecules while preventing larger molecules such as DNA and the proteins. Nucleus of the typical mammalian cell has about 3000 to 4000 pores throughout its envelope. So again in this diagram you can see the inner leaflet, the outer leaflet of the nuclear envelope. Here the fuses, in the center you can see central pore complex, which is formed by the cytoplasmic ring, the nuclear baskets, and the cytoplasmic filaments. So these are the different parts of the nuclear pore proteins which are forming the nuclear pore complex. So next is the chromatin. The chromatin, in the interface, the chromosomes are present in highly extended nucleoprotein fibers called the chromatin. Chromatin is a complex of DNA and the proteins called histones that make up the chromosomes, and in each unreplicated chromosomes contain a single continuous DNA molecule. The mitotic chromosome represents a highly condensed structure. So there are the different levels of the chromatin organization. The structure of the chromatin varies considerably as the cells progresses through cell cycle, and changes in the structures are required to allow the DNA to be used and managed. So lowest level of the chromosome organization is that the DNA and the histones are organized into repeating subunits called the nucleosomes. They are giving the appearance of as beads on a string structure. So nucleosomes, what are the nucleosome? The nucleosome, the core particle, consists of 146 base pairs of the supercoiled DNA. So here you can see these are the supercoiled DNA containing about 146 base pairs of molecules, and they almost wrap almost twice around the disc shaped complex of 8 histone molecules. So these are different histone molecules called histone octamer because consist of 8 histone molecules, and this DNA it coil around the histone molecules and has the two coils around it, and this is the H1, which is the linker histone proteins. So histone core consists of the two copies of histones, the H2A and H2B, and then H3 and H4 assembles into an octamer. So these are the different copies of the different histone proteins which are forming the histone octamer. While H1 histones, the linker histone resides outside the nucleosome and binds to linker DNA that connects one nucleosome to another. So this is the H1, this is the linker histone, and it is attached to the DNA, and it attaches different nucleosomes to each other, thus giving an appearance of strings on beads on a string. So this is again another diagram in which you can see the coiling of the DNA molecule around the histones octamer, and this one is the linker DNA, which is H1, and is attached to the different histones nucleosomes. This is another structure of the nucleosome and another structure of the nucleosome. So in this you can see how the nucleosomes are formed and how they are attached to each other. Again, nucleosomes particles connected by short strands of linker DNA. These are the different nucleosomes particles you can see, and here you can see a short strand of the nuclear linker DNA which is attaching the different nucleosomes to each other. So what is the higher level of the chromatin organization? The chromatin appears in interface cell as tiny dots and fibers of 30 nanometer thickness. The 30 nanometer fibers gather into large supercoiled loops of thick filaments fibers which normally spread through the interface nucleus. So DNA coiling, DNA is tightly coiled structure. This coiling occurs at several levels. DNA is wound around a histone protein core to form the nucleosome, as I have already shown you. About 140 to 150 DNA bases are wound around each histone core, and then 20 to 60 bases form a spacer element before the next nucleosomes. So nucleosomes in turn form a helical solenoid. Each turn of the solenoid includes about 6 nucleosomes. Again another MCQ as well. So the 6 nucleosomes form a helical structure called the solenoids. I will show you this in the diagram in the next slide. So solenoids themselves are organized into the chromatin loops, which are attached to the protein scaffold. So each of these loops contain about 1 lakh base pairs or 100 kilobases of DNA. The end result of this coiling and looping is that DNA is only about 1 by 10,000 as long as it would weigh if it were fully stretched out. So in this slide you can see that this one is the nucleosome, another nucleosome, another nucleosomes. These nucleosomes are coiled to form the structure called the solenoid. So high level of the histone H1 and no gene transcription, in which that you can see the charge N terminal that bind DNA on the neighboring nucleosomes. Here you can see this is highly acylated core histones. These are different the histone particles, especially H3 and H4. There is reduced level of H1 and gene transcription is possible over here. So this is the structure of the solenoid, which is formed by the nucleosome. In this also you can see a beads on string appearance of the nucleosomes, which are attached to each other by the linker DNA, and here you can see the condensed form of the chromosome. In this diagram, you can see this is the double helical structure of the DNA. This double helical structure wrap around the histone particles, histone octameres twice to form the structure called the nucleosome. These nucleosomes are attached to each other by the linker DNA, and then these nucleosomes coiled and form the structure called the solenoid. This solenoid then again coils to form the chromosomal structure and form part of the chromosome. So this is the lowest level of the organization of the chromatin, and this one is the highest level of organization of the chromatin. So what are the different types of the chromatin which are present in non-dividing cell? There are basically two types of the chromatin which are present. This is the hetero and the euchromatin. So euchromatin is lightly packed form of the chromatin that is rich in gene concentration. This is basically active chromatin and is often under active transcription. It is found in both eukaryotic and the prokaryotic cell. This part of the chromatin which is less condensed, and it is responsible for transcription and translation of the genetic material. Heterochromatin is tightly packed form of the DNA, and heterochromatin is inactive and remains compact during interphase. Heterochromatin plays a role in gene regulation and protection of the integrity of chromosome, attributed to the dense packing of DNA, which make it less accessible to the protein factors that bind the DNA and its associated factors. So another type of the types of the heterochromatin which is inactive, again it has two types. One is the constitutive, other is the facultative. So constitutive remains compact in all the cells and at all times, and occur around the chromosomal centromere and telomere. So these are the two locations where the constitutive heterochromatin is present. It remains compact over here in the region of the centromere, and in the region of the telomere. The telomeres are ends of the chromosome where this is present, and it represents the area of the gene with the poor DNA sequence. Next is the facultative heterochromatin. It is the chromatin that has been inactivated in specific types of the differentiated cells, but it is also important that it can be activated at any time. And examples of the facultative heterochromatin is X chromosome inactivation in the female mammals, which is forming the bar body. One X chromosome is packaged in facultative heterochromatin and silenced. It is forming the bar body, while other X chromosome is packaged in the euchromatin and it is expressed. So these are the types of the heterochromatin. One is the facultative heterochromatin, and other is the constitutive heterochromatin. So what are the functions of the chromatin? They package DNA into a smaller volume to fit in the cell, strengthen the DNA to allow mitosis and meiosis, and serve as a mechanism to control the expressions. Human chromosomes are physical carriers of the genetic information, and the packing of DNA into chromosome involves several orders of DNA coiling and folding. So this is the structure you can see in this diagram. This is the inactive cell and this is the dividing cell, in which you can see the chromosomes which are present at metaphyseal plate, and here you can see the condensed form of the chromatin. In this slide, you can see the chromosome and its structure. This is the centromere, where you can see the kinetochore is present. This one is the short arm, also called the P arm. This one is long arm, also called the Q arm. These are the sister chromatid, and this is the replicated chromosome. And this is the chromosome having the P arm, the kinetochore, and the Q arm. So last is the nucleoplasm. It is highly viscous liquid that surrounds the chromosome and nucleoli. Many substances such as nucleotides necessary of purpose such as replication of DNA and enzymes which direct the activities takes place in the nucleus and are dissolved in the nucleoplasm. And the nucleolus, this is the prominent structure in the nucleus. As the nucleolus produces ribosomes which move out of the cell's nucleus and take position on the rough endoplasmic reticulum, where they are critical in protein synthesis. So this nucleolus is basically non-membranous part of the cell nucleus. The cell nucleus may contain one nucleolus. Cell nucleus may contain many nucleoli depending upon the activity of the cell. The cells which are highly active, they can contain many nucleoli as well, and this is the area where basically ribosomes are synthesized and tRNA and messenger RNA are formed, and they take the signal from the cell nucleus to the cytoplasm. So here are the references. These are Basic Histology by the late Ghassan Siddiq, Histology Text and Atlas by John Quera, and Histology Text and Atlas by H. Ross. Thank you very much, dear students, for your attention for the today's class, and Allah Hafiz till the next class.